In this module, we'll talk about the nature of bond which is formed between two carbohydrate monomers. So let's look at this. As we talked about in previous module, the glucose atom, when a glucose molecule, when it becomes a circular molecule, it can adapt two configurations, the alpha and the beta configuration. So when two glucose molecules in alpha configuration form a bond, they have a very different chemistry than when a bond is formed between a beta and an alpha glucose. For example, the starch or glycogen, a molecule which animals and plants use to store glucose molecule, it has glucose molecules linked in alpha configuration, whereas cellulose, a material which is present in the wood, in this material the glucose molecules are linked in beta bond. So let's look at this bond, how this forms. Let's look at this bond in more detail. Here we have on our screen alpha glucose molecule, and we have a beta glucose molecule in the upper portion of the slide. I'll point this out. This is alpha glucose molecule, and next to it is a beta glucose molecule. We have already looked at the structure of alpha and beta, how they are different. In one case, in alpha glucose, we said the hydroxyl group is pointing downwards. In beta glucose, the hydroxyl group is pointing upwards. So when this bond is formed between alpha and beta glucose, a water molecule is removed, a condensation reaction. We have talked about that in our previous module. A water molecule is removed and a bond is formed between two glucose molecules. Now, the glucose molecule on the left up here, since this molecule was in alpha configuration when it formed the bond with the adjacent glucose molecule, this bond will be called alpha 1,4. It is called alpha 1,4 glycosidic linkage. Glycosidic linkage is basically the name of the bond between two carbohydrate molecules. Like we talked about when we were discussing protein molecules, we talked about the bond between two amino acid molecules is called a peptide bond. Here the bond between two carbohydrate monomers is called glycosidic bond. We have talked about bond between alpha glucose and a beta glucose. Now let's look at a bond which is going to be formed between two beta glucose molecules. That is present in the lower part of your screen. Here is a beta glucose forming a bond with another beta glucose molecule. It's the same process. A water molecule is going to be removed. It's a condensation reaction, and a bond will be formed between these two glucose molecules. In this case, since the glucose on our left side, this glucose was in beta configuration, now this bond will be called beta 1,4 glycosidic linkage. What is the difference between these two molecules? In biology, the difference between these two molecules is very significant. The alpha 1,4 bond can be broken down by our enzymes. However, beta 1,4 bond is very stable and we cannot break this bond. That is why we cannot digest plant materials which have beta 1,4 bond in them. Wood, for example, has beta 1,4 bond. Many grasses and many plant structures also have beta 1,4 bond. Let's look at the consequences of alpha and beta bonds in the polymers. Cellulose is also polymer of glucose molecules. In this case, the glucose molecules are linked by alpha 1,4 linkage, glycosidic linkage. As I've mentioned, it is very stable and it is very hard to break this bond. Glycogen and starch, they're both similar molecules. There's relatively more branching in glycogen as compared to starch. These molecules have alpha 1,4 glycosidic linkages. You will also notice there's branching in these molecules, in glycogen molecule depicted on your screen. This branching is a result of alpha 1,6 bond. I will highlight that bond on the screen. Here's the alpha 1,6 bond. This allows this carbohydrate polymer to form branches and makes it more efficient to store energy. Now let's look at the graphic difference. Next time when you see wood, you will realize that wood is made up of same material, same subunits as the roti you eat for lunch or dinner. The only difference is the type of bond between the two monomers. In wood, it is beta 1,4. In roti, it is alpha 1,4.